And we are on. So, reviewing the hypothalamus, just to give you an idea, if I were lecturing to you in person, I'd be drawing many more pictures than I do in these meetings, just because I have more time with you. So, but just to give you a quick review of anatomy of the brain itself, I'm going to draw a profile. It's a frontal, or actually a mid-sagittal section of the brain. So what we're doing is, in layman's terms, we're looking at the brain from the side, the inside of the brain. And if I do that, here's the cerebral hemisphere, here's the frontal lobe in this direction. So this is anterior, this is posterior. I'm just going to give you a quick review on the anatomy, the general anatomy of the brain. The cerebellum is down here in the back, the posterior. I've made it a little small. And the brain stem does this and merges with the spinal cord. The area of the hypothalamus is in the diencephalon. So generally, all this outer region up here is, and it's also the covering of the brain, but if we were to slice the brain in half, a right side and a left side, and we're looking at it, the diencephalon region is underneath this dotted area that I've sort of encircled here. I'll draw it in a different color. So the diencephalon would be everything contained sort of in this interior portion of the brain that we can't see from the outside. And within there, there's the thalamus, the hypothalamus, pituitary gland, the epithalamus. And just because I'm talking about the hypothalamus right now, first I'm going to draw the thalamus are these sort of two large egg-shaped structures like this. There's one on the right, one on the left, and they're connected to each other. They're not separate. It would sort of be like if you made a fist with your right hand and made a fist with your left hand and then brought your two fists together to touch. That's sort of what the thalamus looks like inside. And the hypothalamus, I'll just arbitrarily choose blue. Hypo means below, so the hypothalamus sort of hangs down or is an area of tissue below the thalamus. So I'm just going to put a T for thalamus, and then the hypothalamus is in blue like this. And it comes forward and sits below the thalamus, and it makes this weird shape at the bottom that kind of looks like a W. There's the hypothalamus. That's where it sits in our brain. And I'm just going to quickly draw the pituitary gland. There's actually hypothalamic tissue. This stalk of tissue is called the infundibulum, and the pituitary gland hangs from the infundibulum like this. So this would be the pituitary. I'm going to talk about the hypothalamus today because it's so important to almost every function in the human body. So that's a quick picture of where it exists. Now this structure, I'll go back to blue since we're talking about the hypothalamus. That's what I'm going to focus on for the next 10 minutes. It contains nuclei. And just to remind you, multiple nuclei, just to remind you what that means. It doesn't mean the nucleus of a cell. These nuclei, we defined it earlier, are collections, clusters of neuron cell bodies in the central nervous system. So largely the cerebral hemispheres are also made up of just clusters of nuclei, but the hypothalamus has very specific separate nuclei within its tissue. This organ is considered both a neural organ and an endocrine organ. So we say that it's the major neuroendocrine organ in the human body. It's dual because there are neural cells, the nuclei are the neural cells, and there are endocrine cells that actually secrete hormones, synthesize and secrete hormones. And it does both. It's a dual organ. This is also, this organ, the hypothalamus, is the main visceral control center. So this neuroendocrine function makes it the main visceral. Visceral just means inside, what's going on inside of us. Think of our organs. Think of all your digestive organs, your blood vessels, all that business. This one area of tissue in the brain is the major control center of our visceral insides. And that regulates the main visceral control center, makes it the main regulator of homeostasis. Remember that word that we mentioned the very first day, I think, of class. Homeostasis means maintaining a stable internal environment. So all of those functions that go on inside of us, those millions and millions of functions every second of every day to maintain our insides, to keep them within stable limits, this guy right here is the main regulator of most of that stuff. So it controls the autonomic nervous system. I'm going to talk more about that probably tomorrow and or Wednesday. But it controls the autonomic nervous system. Think of things that we don't have conscious control over. By controlling the brainstem, so the hypothalamus here, it uses this area. I'll use black. Here's the brainstem. There's 3 sections to it. And the hypothalamus is the control center, but it uses these areas of the brainstem then to send signals out to our body, like this. And it controls things, autonomic nervous systems. Think of things, when you hear that autonomic, think automatic, and think of things like blood pressure, heart rate, digestion, respiration, reflex actions, the pupil of our eyes. The list is long. I always tell students, think blood pressure, heart rate, digestion, respiration. Those aren't the only things that it controls, both the brainstem and the hypothalamus, but think of all of those automatic things like those. Now the hypothalamus is also the heart of the limbic system. So I'm still talking about hypothalamus. The limbic system is the area or part of the brain that controls, it's what's called our emotional part of the brain. So the hypothalamus largely also controls our emotions. The hypothalamus is at the root of all of these things that I'm listing. What emotions, things like pleasure, pain, fear, anger. So I just wrote rage. Biorhythms, things like sex drive. All of these things are controlled by everything in blue that I've drawn. I need to go on to another page. Don't worry, I won't erase this. I'm just going to layer over the top of it. So if you want me to go back to it for reference, just ask and I will. I'm still going to continue talking about hypothalamus. Remember, this is where it's located, in the center of the brain. So let's layer on top of this. So we said that it controls the limbic system, the emotional part of our brain. It also regulates body temperature. And it's called the body's thermostat. So when we get too cold, the hypothalamus triggers us to do something to create heat. If we get too warm, the hypothalamus triggers something for us to release heat or initiate cooling. So usually cooling is initiated in the form of sweating, and something like shivering, all those muscular contractions of shivering, that generates heat when our body temperature gets cold. The hypothalamus also regulates hunger. So the feeling of hunger that you have when you haven't eaten in a while is generated in that center system of the brain, that neuroendocrine gland called the hypothalamus. It also regulates our fluid balance. So I'm just going to write water balance, H2O balance, and thirst. When you feel thirsty, that feeling is coming from the hypothalamus. So our thirst centers are located in the hypothalamus. The hypothalamus also triggers, it secretes and releases a hormone called ADH. That stands for antidiuretic hormone. That's not supposed to be, that's supposed to be a D. What ADH does is this signals our kidneys to reabsorb more water. Think of it this way. It tells our kidneys to decrease urination. The reason, here's the deal. When we are dehydrated, we haven't had fluid intake for a long time or for a while, we not only feel thirsty, which is a feeling that comes from the hypothalamus causing us to intake more fluid, but the hypothalamus also triggers the release of this hormone, ADH, antidiuretic hormone, and that tells the kidneys not to urinate as much. That conserves fluid because we're dehydrated. It's a life-saving this is a life-saving mechanism, and so is this, the release of ADH. Because fluid intake is almost necessary, constantly necessary in order to maintain homeostasis. Hypothalamus also regulates our sleep cycles when we are asleep and awake. And when I talk about that, when I mention the epithalamus at the end, I'll mention what organ the hypothalamus regulates to do that. And finally, the hypothalamus basically regulates our entire endocrine system. And how it does that is by secreting things called releasing hormones and inhibiting hormones. It's very complicated, but I'm trying to make it simple, that control the pituitary gland. So we used to think that the pituitary gland was the master endocrine gland because it secretes all sorts of hormones. Lots and lots of hormones, right? So everyone thought, oh, the pituitary gland, that's the major endocrine gland. But then later what anatomists, what scientists figured out was it's actually the hypothalamus that controls the pituitary. So the hypothalamus controls the pituitary by secreting these classes of hormones onto the pituitary to tell it what to do. That's how it is the major control center of the endocrine system. All right, I'm going to say that's it on the hypothalamus. I'm just going to say one thing about the epithalamus. I'm not going to expect you to know really where the epithalamus is. It's kind of picky. Just to go back to this picture, if we're facing this direction, the epithalamus is located posterior to, sort of in this area back here, posterior to the hypothalamus and around the thalamus. That just gives you an idea of where it is, what I'm about to write. The epithalamus, I'm just going to expect you to know it contains a gland called the pineal gland. I learned it to say pineal, but I think that's incorrect. So my teacher who taught me always said pineal, but the correct pronunciation is pineal gland. it got its name because it's pine cone shaped, it's cone shaped. This gland secretes a hormone called melatonin. I'm sure you've heard of it. That's a hormone that helps regulate our sleep cycles, our diurnal cycles, when we're tired and when we're not tired. The hypothalamus actually controls the pineal gland. So the hypothalamus is the monitor of when it's light out and when it's dark out. And then the hypothalamus monitors that lightness and darkness, and it controls the pineal gland to secrete this hormone, which then actually makes us tired. All right, that's it on the epithalamus. I'm going to move on to, I think it's section 12.4 in your book, but I'm going to move on to the brain stem. So let's just go to black. The brain stem gets its name exactly because it looks like a stem. I go back to my first picture. That's what I've drawn here in black. This area is the brain stem. It's not considered part of the spinal cord yet. It merges into the spinal cord at the bottom of the brain stem. But this area that I've encircled in black is the area of the brain, the entire brain stem. And it includes 3 major areas. Okay, so I'm going to go over those. Let me go back to this one first. So I'm just going to put a number 1. And by the way, when I label these 1, 2, and 3, that's the order from top to bottom. So area 1 would be up here, 2 is here, and 3 is down here. Okay, so we're just going superior to inferior when we number these. So number 1 is called the midbrain. That's the area closest to the diencephalon, closest to the interior of the brain itself. The midbrain is involved with things like, I'll mention this again later, but pain suppression. Just going to list a few of its major functions. Fear perception, major eye functions and reflexes. I'm just going to put, I'll put movements and reflexes. What does that mean? So you actually have conscious control over your eye movements. You also have eye reflexes, like your pupil dilating and constricting. And last thing I'm going to write are hearing pathways. Now, the brainstem in general is not loaded with gray matter, is not loaded with a lot of consciousness or conscious processing of information. It's mostly myelinated neural tracts or white matter. So it's mostly just a highway of transmission. So it's a non-thinking part of the brain. So I'm going to label these 3 areas: midbrain, pons, P-O-N-S, and then I'll say some generalized characteristics of the brainstem. The pons, that second area below the midbrain, is mostly myelinated neural tracts. So I'm going to write mostly neural conduction pathways. Just think of connecting highways. These are we call them tracks. Neural pathways, we say, are tracks. And you know that this is white matter. Most of the brainstem in general is white matter, but I'm just reiterating that. And this pons connects the midbrain to the 3rd section called the medulla oblongata. That's a mouthful. So most people, when they're speaking, just say medulla. So the medulla oblongata is mostly also neural conduction tracts, white matter. And it connects with the spinal cord. Also in the medulla, we have, this is an area, I'm going to highlight, this is an area of crossover. What that means is, remember before when we were talking about the cerebral hemispheres, we said the left side of the brain, the left cerebral hemisphere generally controls the right side of the body and vice versa. Well, this is where that area of crossover occurs of pathways. So signals coming, for example, from the left side of the brain, the left cerebral hemisphere, where they cross over to the right side of the body is in the medulla. So this is the area of crossover. I'm just going to put from left to right. That's where that occurs. Also, I have to go to a new page because I'm running out of space. I'm still talking about the medulla here, okay? So I'm going to go to this new page. I'll just, so you know this is the medulla oblongata continued. This has 3 major control centers in it that we do not have conscious control over. It's the autonomic nervous system. I'll write A, B, and C. A is the cardiovascular center. They say centers because there's a specific cardiac center and a specific sort of vasomotor center. But this area controls things like, I'm going to abbreviate this, heart rate, something called stroke volume. Just think of beating power, how hard the force contracts. So SV is stroke volume. That just means how forceful each contraction is. And then the vasomotor centers control things like vessel diameter. When our blood vessels constrict or dilate, that is all regulated, coming basically from the cardiovascular centers. That's supposed to be a C. There's another section in the medulla called the respiratory centers, and you can guess what that does. The respiratory centers or center controls things like breathing rate and how forceful or how deep a breath is. So I'm just going to put plus depth, how deeply you breathe. And finally, there is a third, it's actually a group of centers that regulate the reflexes, things like vomiting, when you get the hiccups. It's coming from a neural center in the medulla. These are called our deglutition centers, but I'm just going to write swallowing. That's a layman's form of deglutition. Coughing. Our deep coughing reflex is regulated in the brainstem at the medulla. And finally, things like sneezing. So all in all, that brainstem controls all of our autonomic behaviors and functions associated with survival. So we say the brainstem, all 3 of those sections, regulate the autonomic nervous system, our basic physiology, survival mechanisms. Things like heart rate, digestion, breathing, and all the things that we've listed. So some people call this area of the brain the most primitive area of the brain. meaning evolutionary time. Other animals have a similar area to their brain called the brainstem. No animals have as developed area of the brain like up in our cerebral hemispheres, particularly our frontal lobe. All right, let's move on now. So I'm going to go to the next chapter, 13.1, and we're going to finish up talking about sensory receptors today. It'll probably be a relatively short lecture today, which is good. I'm just going to write chapter 13. I believe it's 13.1. That makes sense where we're starting. And it's about sensory. I don't have time to go into all the detail. I'm going to try to make it simplified for you. The whole sensory pathway, that afferent part of our nervous system, is what we're talking about. And we know that we start with sensory receptors, both visceral and somatic, but we need to be a little bit more detailed than that in our descriptions. So this part of our nervous system receives information from, I'll talk about these in detail in a minute. It's good that you hear these terms twice. Exteroceptors, proprioceptors, and interoceptors. I'm going to write details about these in a minute. Don't worry about what, this is just telling us where in the body these sensory receptors are located, okay? I'll write their details down in a moment. But first we need to talk about levels of integration. Don't worry, that's just a complex way of saying the pathway of neural stimulus. So you stick your fingertip on a hot stove. Very first of this levels of integration, there is the receptor level. Okay, that just means in your finger, there are sensory receptors that get stimulated from the heat of that hot stove. So that's step one on level of integration. Then two, your book calls it a circuit level. That's just a fancy way of saying the pathway from your finger to your brain, to your central nervous system. So we write circuit level, that's C-I-R-C-U-I-T. This is just the, I'm just going to write, this is just the afferent pathway to the central nervous system. And finally, as always, there's a perceptual level that occurs in the central nervous system, particularly in your brain. So I'm just going to put processing begins. Processing begins in the brain, specifically in those cortex areas. Think somatosensory cortex areas. So this is the pathway. These are the levels of integration. We go from finger on a hot stove, that stimulates sensory receptors in your finger enough to fire action potentials. Those use this pathway to get to the central nervous system. And then once those signals, those action potentials reach their destination in the brain, your brain senses, heat. That's what's going on at the perceptual level. Okay. Now, I'm going to just talk about processing at the receptor level. I'm not going to talk a lot about the circuit level, by the way. So this is at the very beginning, the receptors that are in your finger. Processing at, I'm using that finger on a hot stove example because it's easy for us to imagine. We've all done that before. This could be any sensory pathway coming from outside of your body, sensing touch, heat, pain, whatever, or from the inside of your body, the visceral pathways, sensing digestion, blood pressure regulation, if you have a stomach ache, yada, yada, yada. So this is just talking about a general pathway. Now we're talking about the receptor level, step one, when a receptor is stimulated. And so there's some requirements that must be met in order for that receptor to be triggered, to fire. So first, the stimulus must match the receptor specificity. That's just a fancy way of saying the change, the stimulus, for example, heat in our example, it must match the receptor specificity. It must stimulate, guess what? Heat receptors. That's easy. It's easy to understand. We don't want to, we're not necessarily stimulating touch receptors in our example of hot stove. We're stimulating heat receptors. Secondly, the stimulus must also be within the receptor's receptive field. It must be in the receptor's, what we call the receptive field. Don't think too long and hard about this either. It just means this. If your temperature receptors in your finger can only, quote, I'm putting this in air quotes, measure or be stimulated by temperatures, say, between temperatures from 70 degrees to 5,000 degrees. Well, let's be easier, from 70 degrees to 300 degrees. If the temperature is outside of that receptive field, that receptor will not fire, will not be triggered. For example, if you touch something at 60 degrees, your temperature receptors in your finger aren't going to sense much of a change in temperature. That's all that sentence means. The last thing is the stimulus energy must be converted to a graded potential. This is called transduction. So this is key. So the stimulus must be at a level to cause a graded potential, aka a localized change in membrane polarity, okay, in membrane charge. And then what we call this, the stimulus being causing a graded potential, that has a name. It's called transduction. And I'll tell you why after I write the word. Think of transduction as this, the process of turning some sort of environmental stimulus into a neural electrical response. Okay, so that's what we're doing here. We're turning temperature, heat, into an action potential to fire a stimulus to our brain. This also goes on in all of our sensory organs. For example, our eye converts light waves, which are in the environment. I'm just giving you other examples of transduction. Light waves in the eye get converted to neural impulses at the retina. Our retina in the eye converts light wavelengths in the atmosphere to electrical impulses. Likewise, last one I'm going to give you, sound waves in the atmosphere get converted to action potentials in our inner ear. Actually, what happens is there's more of a conversion. Sound waves get converted to vibrations, but then those vibrations get converted into, guess what, an electrical stimulus. All of these must be met, and all of this is a process of transduction in order for our brain to sense any sort of change. So sensory neuron, also finally last, I've done this transduction, and the last condition that must be met is the sensory neuron must reach threshold to fire an action potential. That's sort of a given. So the sensory neuron must reach threshold from that graded potential. I'm just going to say to fire an action potential to the brain. So all these conditions must be met in order for a signal to actually get to our brain, for our brain to sense it. I'm going to talk about, I'm still talking about processing of sensory information, but this word I do expect you to know, adaptation, because we've all experienced this. What this term means, that's supposed to be an N. Adaptation is defined as a change in receptor. After I read out the definition, you'll know if I give you an example what this means. You might know what it means already. A change in receptor sensitivity. Sorry about the misspelling. Sensitivity in the presence of constant stimulation. So we've all experienced this. When you say your senses, you get used to something, is what that means. Classic examples. You walk into a room where there's a really strong odor, either a pleasant one like perfume or a foul one like, I don't know, onions, if you don't like onions. After you're in that room for a while, you sense that smell, that odor a lot less than when you first entered that room. That is an example of sensory adaptation. So the constant stimulation on your sensory receptors from that odor, that chemical, you get a reduction in sensitivity to it. Likewise, if you put on a shirt first thing in the morning, you really feel that on your skin. But then very quickly you forget, your brain forgets that you're even wearing clothes. That stimulation of the constant rubbing of your shirt or your pants or whatever against your skin, that's an example of adaptation. It's a change in receptor sensitivity in the presence of constant stimulation. Not all receptors exhibit this. Not all sensory receptors in the human body exhibit sensory adaptation, only some. I could give you a million examples. You put a baseball cap on your head, you wear it for 2 or 3 hours, you take it off, and guess what? It feels like you still have it on your head. That's an example of sensory adaptation as well. The next pathway is that circuit pathway. I'm going to skip that. I'm just going to remind you, this just refers to after sensory receptors are fired, the circuit pathway is just a pathway of sending the signal, that afferent signal, to the brain. And I am just going to talk a little bit about then perception. It's way too complicated to talk about all the types of perception that we have in the brain. What I want you to focus on for our last bit that we have time to talk about is the perception of pain, because it's so common and it's what many students are curious about. So I'm going to just talk about perception of pain really quickly. Pain receptors, and there's different kinds, are activated by different types of stimuli. I'm going to just list a few of them. things like pressure, you know, temperature from our hot stove example. Chemicals, I'm going to put a star next to this one. Chemicals released by tissue damage. So if something pushes on you too hard, it can become painful, or knocks you too hard. Somebody whacks you with a hammer or a baseball bat, you hit your finger with a hammer, that is this one. Temperature, burning your finger on a stove. Chemicals released by damaged tissue refers to things like you cut yourself, scrape yourself, your skin. These are easy ones to think of because we've all done it. Anytime tissue gets damaged, it releases chemicals that cause the perception. It fires receptors to send a signal to the brain and the hypothalamus that you sense pain. We also have on this topic pain. I said I was going to mention these earlier. Our brain makes pain suppressing chemicals as well. You've probably heard of this one, endorphins. These are sort of pleasure releasing chemicals. They cause the brain to sense pleasure. They're released when we have a pleasurable experience. It can be something like a sexual experience or physical stimulation, but it can also be, endorphins are also released from exercise. When you exercise vigorously, you've probably heard of the athletic or the exercise high after an activity. That comes from the release of endorphins. Chemicals can also release endorphins. Certain drugs can release endorphins. There's another class of chemicals that are pain-suppressing chemicals called enkephalins. That's ENKEPHALINS. Enkephalins are inhibitory chemicals that they physically dampen pain signals in the brain. So they're less pleasurable chemicals. These are pleasure chemicals that sort of mask pain, and enkephalins are specific inhibitory chemicals that actually dampen those pain signaling pathways. All right, let's try to get to the end of this. Sensation and perception of stimuli occur in the brain. We know that. So you cut your finger. The actual pain isn't, we think it's at the finger, it's from tissue damage, but the sensation of pain and all other senses, so I'm just putting sensations in general, occur in the brain, not at the point of stimulus, not at the hot finger on the stove. That just sends a signal. It's actually in the brain that we say, that's painful. And now I'm going to do some receptor classification types to end this discussion. Your book goes through several different classification types. I'm going to try to be quick and simple. First, we're going to look at by stimulus type. Okay, there are mechanoreceptors. These get stimulated by things like touch, pressure, stretch. Yes, stretching in your muscles, which is what you're probably thinking, but also stretching of like vessel walls and organ walls. There are mechanoreceptors in our vessels and organs, or things like vibration. There are also thermoreceptors. That's an easy one. Those are temp receptors. Just think changes in temperature. There are photoreceptors in the retina of the eye that are sensitive to light wavelengths. There are chemoreceptors. These are a little bit more complicated. These sense changes in chemicals, both in the atmosphere, so things like smell and taste, or change in body fluid chemistry. We have things like oxygen and CO2 receptors in our bloodstream that sense the levels of certain chemicals in our blood. So they're both inside and outside. And finally, nociceptors. These are pain receptors. Okay. Now I'm going to quickly classify receptors by location. So I'm going to put a number 1 here by stimulus, then number 2 by location. This was the first set of receptors that I wrote down at the very beginning of this conversation. Exteroceptors monitor the external world. So we find these around body surfaces. Interoceptors, the name gives it away, they monitor the internal environment. And we find these receptors, these are by location, on the inside of the body. And finally, proprioceptors. These are found internally, but they are specifically found, so I'm going to write this one out, specific in skeletal muscles, tendons, joints, and ligaments. And these monitor movements and body position. So you're sitting up in a chair right now, I assume. If you start to lean to your left too far, you'll fall over. Proprioceptors in your body constantly monitor body position. And those are the things that fire, believe it or not, even when we fall asleep sitting up. You've seen somebody fall asleep sitting up and then they jerk to right themselves up. Those are proprioceptors firing to the brain. And finally, I'm going to do number 3, classification by structure. And this is our last topic of the day. So we can classify receptors by stimulus type, and there are all the different types of receptors. We can classify receptors by location in the human body, and there are the types of receptors. And finally, by structure, physical structure, what they are made of, what they look like. This is complicated. I'm not going to get into all of the detail probably today, so I'll start this and then continue in tomorrow's lecture. So there are something called simple receptors. Sometimes these are called general receptors or general sense receptors. And what they are, how they're made up, is They are non-capsulated nerve endings. And they typically have, we call them free nerve endings. They have little knob swellings on the end of them. These tend to be just dendrites. I'm just going to write dendritic and uncovered, non-capsulated dendritic ends is what they are. They're simple receptors. They have a little tiny knob on them. So think of this. Remember in our neuron picture, our motor neuron picture, think of a sensory receptor, which is really, I should draw a bipolar neuron is what I should draw. Or a unipolar neuron, like this. This dendritic end is the sensory receptor itself. So we have touch receptors in our skin that just this dendrite, if I magnify it, it kind of looks like this. It's just a coiled dendrite nerve ending, and it has a little bulb on the end of it. When that is stimulated, it causes a change in ionic polarity, the graded potential. If it reaches threshold, that fires an action potential to the brain. These are the most common, most numerous, in the human body, and they respond to all sorts of things. Temperature, pain, some pressure. We have other deep pressure sensory receptors. This is more light pressure, surface pressure, and something like itch. When you get an itch in your skin, it's one of these simple non-capsulated nerve endings that's being stimulated. That's usually by a chemical change, by the way. So we've got non-capsulated simple receptors, and I'm going to end up with, I'm still on number 1, okay? So I'm just going to go to a new board, still under number 1, and just write encapsulated. These are also a form of simple receptors. And it just means that the nerve ending is encapsulated in some sort of connective tissue. Has the basic same structure, but it's in this little encapsulation. I'm going to write CONN tissue. Almost all mechanoreceptors are this type. So you don't even have to write almost. All mechanoreceptors are this type. That means things that are sensitive to pressure, touch, stretch, things like that, some sort of physical change stimulation. Deep touch would be these mechanoreceptors, pressure receptors. Okay, guys, I'm going to stop there for today. If I have anything else to say about this, these classification by structure, these dendritic endings of sensory receptors, I'll bring it up at the beginning of tomorrow's business. So if you have any questions, you can fire away. I'm just going to stop the recording, my recording.